Bismillahirrahmanirrahim. Assalamu alaikum, dear student. Today I am going to discuss structure of bacteria. It is a lengthy but so easy topic. Understanding bacterial structure is a crucial for antimicrobial, that is antibiotic development, and which we targeting specific component of bacteria. Disease diagnosis, in which we identify specific pathogen against a disease, and research development, in which we study the behavior and interaction of bacteria. Basic bacterial components are cell wall, cell membrane, mesosomes, cytoplasm, ribosomes, and chromosomal DNA, while in additional or optional parts are plasmid, capsules and slimes, flagella, pili and fimbriae. Some bacteria may have or may not these structure. Cell wall, it is outermost layer or boundary of the bacterium cells. It is rigid. It is made of peptidoglycan, that is murein, which consists of protein and carbohydrate complex. It maintains the shape. It provides protection to bacterium. It regulates osmotic pressure, in which it prevents the bacterium from osmotic lysis. Next to the cell wall, there is a cell membrane, which is inner to the cell wall. It is thin, delicate and flexible. It have different kind of lipids. It is selective permeable membrane. It control transport of substances in and out of the cells, that is bacterium, and it maintain cellular environment. The cell membrane invaginate and unfolded and form a pocket-like structure called mesosome, in which cell division and replication of DNA occur. Cytoplasm, it is present inside the cell membrane or plasma membrane. It is thick, semi-transparent and elastic. They are jelly-like or gelatinous mass of protein, carbohydrates, lipids, salts and inorganic ion. The semi-fluid jelly-like portion is called cytosols. It lacks membrane-bounded organelles. The metabolic process occur inside the cytoplasm. Ribosomes, inside there are many ribosomes. These are teeny granular substances. They are freely dispersed in the cytoplasm. They are smaller in size, that is 70S. Mainly it consists of RNA and protein, so-called ribonucleoprotein. It helps in the protein synthesis. There is no prominent nucleus and membrane bound organelle present inside the cytoplasm. The main chromosomal DNA is present in a specific region called nucleid, which contain bacterial genomes, which is essential for its survival and function. Just like computer, it is the main hard drive in which essential file are present. Plasmid, it is smaller, circular, double-stranded separate DNA in the form of ring. It is called extra chromosomal DNA because it is present outside of the main chromosomal DNA. It replicates independently of the main chromosomal DNA. Just like computer, it is like a external drive or USB sticks in which additional files are present. It carries fewer or specific gene, that is antibiotic resistant, Recently, it is also used as a vector and genetic engineering. Some bacteria secrete extracellular material, that is capsule and slimes. These are collectively known as glycocalyx. These are sticky, gelatinous, viscous covering envelopes or coating of fibers. When these layers are tightly bonded or attached to bacterium cell wall, it is called capsules. When these layers are loosely or less tightly attached to bacterium cell wall, it is called slime. These are made of polysaccharide and protein, that is glycoproteins. It have a main role in adhesion, in which it adhere or attach to host cells. It also help in the protection against desiccation and dehydration, that is drying out. It also prevent phagocytosis by the immune cells, so it acts as a antibiotic resistance. It contributes to bacterial pathogenicity and virulence, which enhancing bacterial ability to cause diseases or infection. Flagella, they are whip-like appendages. It have three parts, that is basal bodies or apparatus, which is associated with the plasma membrane and cell wall. The second part is the short curved hooks, and the third part is called helical filaments. The hook and filament are made up from protein called flagellin. Flagella help in motility on the basis of size and position. They may be monotrichous, in which single flagellum is present, lobotrichous, in which group of two or more flagella are present at one pole or end of bacterium. Ampitrichous, MP means both, in which group of flagella are present at both end or pole of bacteria. Peritrichous, in which flagella disperse on the entire surface of bacterium. Atrichous, the bacterium which have no flagella is called atrichous. Beside these structure, some bacteria have surface appendages which play no parts in motility. These are pili and fimbriae. Pili, singular pilus, it is a Latin word which means hair. These are hair-like. They are larger and thicker. They are few in number, that is from 1 to 10 parcels. Sometimes it help in adhesion, in which it attach to some bacterium. It also helps to transfer DNA from one bacterium to another bacterium called conjugation, and this time it is called conjugated or sex phallus. It is made of protein called pili protein. Pili are present in gram-negative bacteria. Fimbriae, singular fimbria, it is a Latin word which means thread. They are present in gram-positive bacteria and gram-negative bacteria. They are bristle-like. They are shorter and thinner than pili. They are more numerous, that is up to 100 per cell, than pili. They are involved in adhesion to the surface of bacteria or host cells. It also helps in the colonization and biofilm formation. It is made up of protein called fibrillin.